All right, hey everybody. So today I'm gonna talk you through and walk you through the leaf disc, the floating leaf disc lab. So one thing to keep in mind is obviously that we are looking at photosynthesis here, and so we just have to remember the equation. So the equation is plants need carbon dioxide and they need water, and they take that carbon dioxide and water in the presence of oxygen and convert it to sugars or glucose, and then oxygen as a waste product. So what we're using is spinach leaves, and these floating discs are made by hole punching these spinach leaves into these pretty uniform shape, what we call leaf discs. So leaves naturally have oxygen in spaces within the leaf, and that oxygen causes them to float in liquid. So obviously we're talking about that waste product of oxygen in photosynthesis that's sitting in these spaces that make these leaves float on water. And what we're going to do is infiltrate those oxygen spaces and put actually carbon in them by using a bicarbonate solution that we made that was looked at in your procedures in the first 5 steps. So this bicarbonate solution goes in those spaces, it increases density of the leaf, and then therefore makes it sink. So what we're going to do is we're going to sink all these leaf discs, and then we're going to put them in solution and then measure photosynthesis indirectly by looking at how long it takes oxygen to refill those spaces. So the first thing we have to do, I set up everything in the lab, which is on Schoology for you. And the next thing that I have to do is that I have to sink these leaf discs. So the way we're going to do that is creating a vacuum in a syringe. Okay, so the first thing you do in there's 10 leaf discs here, is we're taking our 10 leaf discs, we're putting them into the syringe. And when I put them into the syringe, I'm also going to put that bicarbonate solution, which also has soap on it, which you should have read. The reason that soap is added to this is because the leaf's surface itself is hydrophobic. So the soap, here's the solution, this 3 milliliter solution that we're adding says the soap is kind of counteracting that hydrophobic surface and going to allow this bicarbonate solution to go in there. So if you can see this, where's the camera? These leaf discs, and that one will get down in there. These leaf discs are floating right now. I'm gonna make them sink. And the way that I do that is adding this plunger and creating a vacuum. So we're gonna put this plunger in. I'm gonna get all the air out. And then I'm gonna create a vacuum. Once I create the vacuum, and I know you're not gonna be able to see this super well, so I'm creating a vacuum. I'm gonna swirl this so that these leaf discs eventually get that bicarbonate solution in there, okay? So now we'll see, can you see that? All right, they're starting to sink. I'm gonna just one more time because I already did this, and then a couple started to float right away, which is okay, but we'll just make sure that these are really infiltrated with bicarbonate solution really well. Let that down. Okay, oh, sorry, my hands get in the way. Perfect. Now instead of floating, all of those are sinking to the bottom. That means that they have been infiltrated with that bicarbonate solution. So the next thing I do is I take a cup, sorry, I'm trying to make it so that you can see all this stuff, and I'm gonna dump these into this cup right here. Okay, there's a couple in there that are stuck, but we'll take this solution and get those other ones out, because then we need to add this bicarbonate solution to that anyway, in which they're going to be floating in. So if we look at these leaf discs in here, oh, sorry. Look at these leaf discs in here. We'll see that all of them are now floating to the bottom. What I'm gonna do is I'm gonna put one in light and one in dark, and I'm gonna time it, and every minute I'm gonna count how many leaf discs float or start floating to the top. You can see here I have my light source that I'm gonna put one in, and then I'm gonna do the exact same thing that I did with these leaf discs, and I'm gonna put those into a cabinet in dark. I'm not gonna make you watch these leaf discs float, but I am gonna get you the data. So again, I'll explain the data to you. By the time when we're looking at these floating, which actually I kind of cover up right now, that is gonna show us the rate of photosynthesis in a light environment versus a dark environment, which I'm sure you can guess kind of what's gonna happen. But we'll give you that data anyway, and I'll explain what you have to do next, which is graph it and then answer the questions that are on that lab. So I will also have a document that states the data from this lab, which you will want to look at. And then we'll also have a day to ask questions if you're not totally understanding what's going on, but we'll get there. All right, hope that makes some kind of sense, and I'll get you that data as soon as possible. All right, take care, guys. Have a good one.